Well, good afternoon, everyone. We might make a start while we wait for our moderator to join us. My name is Dale Bailey, and I'm a physicist at Royal North Shore Hospital and University of Sydney. Welcome to the first of this new lecture series where the Better Healthcare Technology Foundation is attempting to break out and have a bit of a more diverse program than we might normally get at our usual chapter meetings and branch meetings. For those of you that don't know, the Better Healthcare Technology Foundation is the fundraising and research support arm of the Australasian College of Physical Scientists and Engineers in Medicine. The foundation's objectives are to promote research and development in medical physics, engineering and the associated sciences, and also the safe and appropriate use of innovative medical technology. The foundation endeavours to achieve this by promoting knowledge exchange between health professionals and within the community, awarding prizes and grants for outstanding achievements and innovative projects, and for supporting training and education initiatives, especially in low to middle income countries and regions. And there's a particularly active group in the Asia Pacific area doing that. This lecture series is aimed at the wider audience of scientists and engineers and intended to provide insights into areas beyond their normal professional domains. It is intended to promote cross-fertilisation of ideas between individuals who may not normally collaborate, and seeks to encourage a multidisciplinary culture for physical scientists and engineers beyond their traditional craft groups. We've got three speakers today, and I'm indebted to Professor Ben Eggleton from the Sydney Nanoscience Hub for suggesting this program today. So he was very much at the origins of this. The three speakers we have, and I think we've had a late reversal in the order, but the first off is going to be Professor Zdenka Kuncic. Zdenka's got a BSc in physics from the University of Sydney and then went on to do a PhD in astrophysics at Cambridge. She's now leading a very distinctive interdisciplinary research program at the interface between physics, medicine, biology, neuroscience, and engineering. Her research focuses on developing and applying physics-based approaches to challenging problems that can really only be addressed with highly multidisciplinary perspectives and strategies. So her area is very much within what we want the remit of this lecture series to be. There'll be 4 lectures throughout the year at regular intervals and at regular times that you'll be able to participate in. So I'm hoping that our coordinator is going to join us very soon, but I think we'll get started with the first lecture. Can I just remind everybody to leave their cameras off and their microphones off, and submit any questions that you have, which we'll take at the end via the chat function. So with that, it's my great pleasure to hand over to Zdenka. Thank you, Zdenka.
Great, thanks very much, Dale, and also thanks very much to Alice, who we're still waiting on, but also to Ben for helping to coordinate this event. So as Dale mentioned, I have been involved in medical physics research for some time now, and I can see a lot of familiar faces online. So my talk today is going to focus on how we can use nanotechnology in medical physics research to actually take it beyond conventional radiotherapy, as it were. So I'm going to be talking about theranostics, but adding the nano at the front of it. And in particular, the nano I'm putting at the front of theranostics is really all about how we can harness the unique functionality of nanoparticles to do more than we are currently able to do in theranostics. So nanoscience and nanotechnology is all about geometric confinement. That's and one thing you take away from this talk, it should be that. Now, what happens when you confine a material down to a nanoscale, let's say less than 100 nanometers, in 1, 2, or even 3 dimensions? There are 3 things that happen. Firstly, you get a dramatic increase in surface area to volume ratio. Secondly, you start to discover some really interesting mechanical, optical, electrical, and magnetic properties that the material does not exhibit on the bulk scale. And thirdly, it's also possible to realize some non-trivial quantum effects. So what I'm going to talk about today is really harnessing this type of unique functionality that only emerges on the nanoscale, and think about medical applications. So that's the first challenge that I'm seeking to address. And the second challenge is how can we maximize opportunity for clinical translation? So it's all very well to be doing research in the lab and publishing papers. But at the end of the day, all of us want to be able to translate this research and really create impact. So I'm going to focus my talk today on one particular type of nanoparticle called super paramagnetic iron oxide nanoparticles, or spions for short. And these are nanoparticles which exhibit extremely strong magnetization in the presence of a magnetic field. So in this diagram, the top panel shows the case for a ferromagnetic particle when there's no magnetic field, and the corresponding nanoparticles on the right-hand side. And the bottom panel shows you what happens when this particle is placed in a magnetic field, and you can see all the magnetic domains with aligned spins. And indeed, that is maintained when you're dealing with nanoparticles of super paramagnetic materials. And so I'm going to be talking about how we can harness this spin alignment functionality. So the most obvious application is in MRI, magnetic resonance imaging. And in MRI, these spions can interact with proton spins in such a way as to shorten their relaxation times. And they induce effectively a really strong nanoscale magnetic field in homogeneity. So in this diagram, this is the nanoparticle, and these are all the water molecules surrounding the nanoparticle. So in particular, spions can enhance T2 or spin-spin MRI contrast by increasing the relaxation rates, and this results in signal quenching. So if we look at this image, this is a T2 weighted image on the left-hand side, no nanoparticles, and on the right-hand side, you can see some regions after nanoparticles have accumulated in these two highlighted areas where the signal has been noticeably quenched. Now, this signal quenching for T2 weighted relaxation depends on the relaxivity of different nanoparticles. And I've been focusing on two types of commercial nanoparticles, one called ferriheme and the other one called precision MRX. And this really speaks to my second challenge, which is how do we do clinical translation? So it's very easy to make nanoparticles in a chemistry lab. There's a lot of people do it. But at the end of the day, we really want this to have meaningful impact. So there's no point in using nanoparticles that have no history of clinical use, because you're going to be very far away from clinical translation. So I've focused all my research in this area on these two nanoparticles, the left-hand side ferriheme, which is FDA approved, and the right-hand side, precision MRX, is a new nanoparticle that's now being commercialized, and the company is now seeking FDA approval. And there's a really exciting clinical translation story that I'm going to tell you about with this nanoparticle. So this particular nanoparticle, we've already tested it in a small animal imaging. So this was done at the University of Sydney. On the left-hand side shows you a T2 star weighted MRI image of a healthy mouse without any spions. And on the right-hand side, you can clearly see delineation in the kidney where, as a result of spion uptake. And in this particular case, the T2 star actually enhances the magnetic susceptibility gradient, which gives rise to the image that you're seeing there. Now, in addition, this particular type of spion that's being developed by a company called Imagion, I'll just quickly switch back here. So that's the company there. We've also tested these spions at ultra low magnetic field strengths. So these are millitesla field strengths as opposed to Tesla field strengths that are used in most clinical MRI scanners. And the reason why we did this is because these ultra low field MRI scanners have now been developed and commercialized. They are portable, which means you can bring MRI to the bedside. They're portable because with the ultra low fields that they operate on, you no longer need bulky superconducting magnets to do the MRI. So with these nanoparticles, we're actually now working with the company to deliver them to this other company, Hyperfine, that's developed these portable MRI scanners to improve the image quality of their scanners. We've also done another study on the other types of spions where we've radio-labeled them with PET isotopes so we can combine them for use in PET MRI, which Dale is obviously very keen on here. So hopefully we'll be able to do some clinical studies in that space shortly. And we've worked with our collaborators at Harvard who have developed this technique for radio labeling this particular nanoparticles without the use of a chelator. And this is very exciting because it's basically opened up a whole new range of radioisotopes, clinically suitable radioisotopes that we can now explore with this nanoparticle for diagnostic applications, but also for therapeutic applications. We've also shown that these radio-labeled nanoparticles, we can actually switch between T1 and T2 contrast in the MRI imaging in PET MRI. And what's really nice here is that because PET MRI combines the high sensitivity of PET with the high resolution of MRI, we've shown that we get an overall improvement in image quality as a result of combining those two capabilities together. Another really interesting thing that we discovered with these radio-labeled spions is that we've shown that the PET resolution in PET MRI can also be enhanced as a result of these radio-labeled nanoparticles. So in this image below, going from left to right and then down, this increasing concentration of the spions in a phantom, and these spions were labeled with a PET tracer, so you can see the gamma spatial distribution in these phantom images, and you can see instantly going from A to D, for example, how much more spatially localized the gamma counts are as a result of the spions in the magnetic field that basically localize the positron trajectories. And extending that work, we've also shown through simulation studies that in principle, these radio-labeled spions can also enhance radiotherapeutic efficacy because of the secondaries that are produced, in particular the Auger electrons, which in the presence of a magnetic field end up producing additional radiotherapeutic effects very close to the nanoparticle and therefore in a much more localized manner than is the case without a magnetic field. So a lot of studies have been done on radio enhancement by nanoparticles in the external beam radiotherapy context, but there's still this idea of then applying this to internal radionuclide therapy is still a very new idea, so we're very excited about this. And I just want to acknowledge all the people who have worked on this and also our funding agency. Thank you very much.
Thank you so much, Zdenka. I apologise to all of you for joining late today. I was having some Zoom issues, and unfortunately, one of our colleagues, Dr. Shelley Wickham, isn't going to be able to join us for this session, and she sends her sincere apologies and hopes to speak to you soon. I'm Alice Motion. I work at the School of Chemistry in the University of Sydney Nano, together with Zdenka and Wojciech. And Zdenka, that was a fantastic presentation. Thank you so much. A lovely talk too. So Wojciech, Zdenka and myself are delighted to join you today for the Better Healthcare Technology Foundation topical lecture. And part of what we wanted to share is just a sort of a taster of some of the areas of medicine that we're working on at Sydney Nano. I know my colleague, Dr. Wickham, would be really happy to present to you in the future. She's doing some fantastic work with a team building autonomous programmable robots, and it sounds like sci-fi, but it's becoming more like reality day by day. So I'm sure that you will hear from her in the future. We'd love to invite some questions from the audience, but I thought perhaps I would start us off, and please feel free to unmute and to show yourself on your camera if you feel comfortable, or to ask a question in the chat. But I thought perhaps I'd start, this is to both of you, Zdenka and Wojciech. I was wondering, as people who for a long while in your career have sort of worked in a very interdisciplinary way, but based within a school, how have your perspectives as researchers changed when you've kind of come together around the idea of nanoscience and nanotechnology? And has that changed your approach or your collaborations in any way? And perhaps I'll ask Zdenka first.
Okay, sure. Thanks, Alice. Yeah, I mean, it's a great question. And for those of you who are not really familiar with, what we do on campus at the University of Sydney, it has traditionally been very siloed. And a number of years ago, our bosses up high decided, well, we've got a lot of intellectual resources on campus, and we could be doing more than what we're doing within our schools and faculties and research groups. So Sydney Nano is one such initiative, and we have a few others on campus as well. And I guess the whole idea behind Sydney Nano is to bring researchers from across the university, no matter what their discipline is, bring them together to help solve some of these really big problems that can only be solved by people with different areas of knowledge, skills, and expertise, all, I guess, with the common theme of nanotechnology. So as I mentioned in my talk, nanotechnology is all about discovering these really new functionalities, new physical properties of materials once they're on a nanoscale. And there's just so much there to uncover. And then the next step of that discovery process is thinking about, okay, well, how can we apply them to some problems? How can we develop new technologies out of the science that's being discovered in that way? The context that we've been speaking about today is really around biomedical technologies and biomedical applications, but of course there are other applications as well. So for me personally, it's been really exciting to have the university back the idea of how important multidisciplinary research really is. And I'm sure those of you who are working in the clinical space already have that appreciation. You guys already work naturally in a very multidisciplinary way. So us academics still have a lot to learn.
So thank you, Zdenka.
So in my situation, Alice, I think it is pretty much similar to Zdenka. I've never published a single paper which was not interdisciplinary. From the very beginning of my career, everything was interdisciplinary. 70% of my publications have the international partners. So this was pretty much in the DNA of my work, of the research and the area in which I work. And establishing the institutes, MDIs within the University of Sydney was the enabler, was something which accelerated and perhaps allowed new connections, and maybe in some cases, moving this to the next level. But in the context of approach, I don't think I changed anything. It is more about expanding and looking into the areas which I was previously maybe missing, because I didn't know that people actually have the expertise in certain areas. So it was more about accelerating and expanding on the research scope.
Yeah, thank you so much. I think a lot of what both of you said resonates with me, even though I'm from a different disciplinary perspective, but the silos that can be present at university, it's quite challenging to break them down. And even through interactions over coffee, bumping into people and finding mutual interest can often we can find ourselves working on the most challenging problems or most exciting problems in that way. Dale, thank you so much for the invitation. I think you have a question for us.
Yeah, I think particularly to Wojciech, many of the audience today will be biomedical engineers, medical physicists, and so on. And I'm just wondering, in your field, where's the balance at the moment between academia driving this or industry? Is this still very much within the universities, this engineering of EVs? I think it's a name you're going to have to change or an abbreviation you're going to have to change pretty soon. But Yeah, I just wonder how far developed is it, and where's it at, and is it about to, sort of leap from academia into industry in a big way?
Look, this is a great question. If we look on the publications and the rate of the growth of the publications, the EV area is actually the fastest growing area in medicine. This is number one, no doubt about this. And the ISEF Society, International Society for Extracellular Vesicles, is implementing really a very strong rigor around the research, reproducibility. We don't want to have the same problem which we have with GMO or stem cell therapies. So there's huge amount of work which is happening at the university, fundamental research. But for the last 5 years, the number of companies which took EV research and started working on this is huge. Eli Lilly, Roche, AstraZeneca, all these companies now investing huge amount of money. And there is probably around 150 startup companies a year in the EV area. In Australia, we have probably about 10 or 12 new companies last year and this year, which are focusing on the EV science. So there is a lot of work what is done around fundamental discoveries in the labs, but there is already a lot of happening in big pharma, and big pharma got really interested in this area. And as I said, the investment from Eli Lilly, $1.7 billion is huge. This was one of the largest deal ever. It was done in the UK with one of the universities.
And just, I'm still sort of trying to understand the EVs themselves. Do you need to have human cells producing them, like we've done traditionally with antibiotics and things, or are they synthetic now?
Fantastic. So they are probably the most common three categories is human cells, bacteria, and plants. So we can actually derive them from anything which you want. We even take them from the fermentation process. By the way, beer contains EVs, but not the mainstream beer, but a craft beer, because filtration will remove them. So look, there is already work which is done in the space of, we call it engineered EVs. So what it means that we actually try to create them, recreate them, but it's impossible. Put it like this. If you start recreating them, it means that you are packaging a few microRNAs in the liposomes. That's the bottom line. But a cell is creating something what is so much more sophisticated that any human invention has never actually seen. So this is still very complicated. And in the process of development of the EVs, there are a few areas which dominate. First is how to produce them and how to produce them at large scale. But the second one is how to isolate them. Every single method of isolation will isolate certain populations or subpopulation, what we call, of the EVs. And the biological function of your, what we call EV-rich preparations will vary of your production, which includes isolation. And these are some bottlenecks which we develop at the moment. There's another aspect, which is regulations. We work very, very closely with the TGA at the moment on some of the aspects, but it is still a little bit difficult in the context of regulatory bodies to classify them. By the way, they are classified at the moment as the biologicals.
Right, thank you.
Thanks, Wojciech, and thanks for giving me a scientific reason for my preference for craft beer. I can cite you now on that one. Zdenka, did you have anything you wanted to add to Dale's question? Or
not in particular, no. I'm happy to move on to other questions.
Okay.
So Alice, can I add, maybe there's something of interest for some of the people on the call. Actually, human milk contains a huge amount of the EVs, huge. And this is an area which is booming, human milk as the therapeutic. And I know the group in the Netherlands, I work with them, they actually come to my lab to do some tests. And there is a huge amount of work, which is how the disease may pass from mom to the babies through the milk which they drink. Normal milk also contains EVs, the cow's milk. I mean, any milk, by the way.
Thanks, Wojciech. We have a question in the chat, which unless Effie unmutes, I will ask for them. But I think Effie says that, and sorry if I pronounce anybody's name incorrectly, feel free to tell me off too. Effie's really interested in the use of nanoparticles in nuclear medicine and would like Zdenka and Wojciech to expand on some specific projects in this area, please. What are the targets? Maybe we go to Zdenka first for this one.
Yeah, sure. So metastasis is a big target. And although I'm not aware of nanoparticles being used to target METs in nuclear medicine, Dale might know, but certainly there's an obvious step in that direction, given that there are now clinical trials being undertaken using nanoparticles in external beam radiotherapy to target brain mets, for example, and other types of metastases from a whole range of different primary tumour sources. So in a separate project that I didn't have time to speak about today, we're working with a company that is about to start phase 2 clinical trials on their gadolinium-based nanoparticles. And as many of you know, gadolinium is currently used as a MRI contrast agent. The industry is looking for a gadolinium replacement because of brain toxicity issues, but for the patient cohort that are taking these clinical trials, that's a secondary concern for them, unfortunately. Now their gadolinium nanoparticles have also been radio-labeled with PET tracers. So again, there's some really exciting research to be done around using those in PET MRI for diagnosing disease. And then, step 2 would actually be treating the disease itself. We've also been looking at radio-labeling the spions that we've been working on for metastases that moves into the lymph nodes. So looking at how we can actually use spions that have been radio-labeled with a PET tracer and using PET to then detect lymph node metastases that would then help in the diagnosis and staging of tumours. So most of these are still very much in the research space. There's a lot of work that would need to be done to move them into clinical trials for clinical translation.
Thanks, Zdenka. Maybe I'll ask a follow-on to that one, if that's okay. So in terms of, you mentioned things that are the things that you're working on are largely in the research stage, but I wondered in terms of communicating possible new therapies or theranostics to patients, whether there's much research being done about patients' attitudes or responses to the use of nanoparticles or nanotechnology for new treatments. And I think Wojciech probably has something to say on that too, but I thought I would start with you if that's okay, Zdenka.
Yeah, I mean, it's a great question. Just the word nanotechnology conjures up all sorts of misconceptions, preconceptions. So the whole communication piece around that is really, really important. That's separate to then thinking about, well, okay, using nanotechnology for new medical technologies, which then are kind of like another hurdle you need to get past. So I think there's a lot more work that can be done in the communication piece. And yes, Wojciech, you probably do have a lot to say about this, particularly around any potential conceptions around toxicity concerns and so on.
I would maybe add, Zdenka, to this, that education is not only a communication, but education and telling the society and telling potential users of these technologies that this is actually life-saving. Sometimes the problem which we have, that the negative story is overwhelming anything which is positive, because we assume that there are hundreds of thousands of patients treated in a positive way is a norm. But if someone was accidentally intoxicated by some of the compounds, it is not. So it will always surface if there is something really negative. So I would say there's obviously a lot of work around the safety, and I think we are in a very good hands at the moment in terms of regulations. The regulations around safety of nanotechnologies or nanoparticles are really, really stringent. So I think whatever gets to the market is really well researched, and it is definitely for the purpose of us, for the sustainability and for the health purposes.
I had another question I wanted to fire at the
Please go for it, Dale, and please anybody else feel free to join in too. Yeah,
while we have this really diverse audience and wide ranging, I think you're fairly familiar with some of the range of activities in the Nanoscience Hub at the university. And I know recently I've been, had a discussion or two from a cancer research point of view with them. And I've been really surprised at the really wide range of things going on. Could you elaborate on some of those, like some of the nanobots and the organ on chips and all that sort of really, like, to me, it's science fiction almost.
Really, really wacky. Well, I think, Wojtek, you know a fair bit about the organ on chip. It's practically, what you're doing at the moment, right? I don't know if you want to jump in there. And
so the model which I presented, when you had the patients, you assemble, I would say in the literature, you will find it is called organ on a chip. We don't like this name, and we call it pathophysiology, human pathophysiology mimicking models. And there are different aspects to this, how you build them, what is your substrate when you actually look at them, whether you use microfluidics, whether you use larger platforms, and all of them have the benefits, and all of them have some disadvantages as well. And it is more about assembling techniques which can answer your question. But it's not only assembling your model, but being able to examine, investigate, and test this model. So what I showed you is just a snapshot of a few techniques which we apply. In reality, we will be applying probably about 20 or 30 different techniques to have a confidence that what we are doing is working properly. So it's also building capabilities which are coming together as the integrated tool to answer our questions. And this is the strength which we have here, because it is not only creating this model, but also being able to examine these in a way that can answer the questions which we ask.
Yeah. And I was just going to maybe add, so for those of you who aren't super familiar with Sydney Nano, please do head across and have a look at our website, and you're very welcome to come to our events. And if you're part of the uni, you're very welcome to join as members. But there's different types of membership, and I forget some of them, but they're associate memberships too for those folks who aren't affiliated with Sydney Uni. And we're always looking for new collaborative partners, so please feel very welcome to join any of our events. We've actually had 6 inaugural grand challenges, of which Zdenka, Wojtek, and Shelley are leaders from 3 of those 6 grand challenges. And you've heard a little bit about the work that's just some of the work that's emerged from these projects and expanded beyond them. But we have 4 brand new grand challenges that have just started at the beginning of this year. And 2 of those are in the health space, although you could argue that all of them are linked to health. So one is looking at solar fuels, and one of them is looking at buildings and cities of the future, which I think both of those areas are linked to health, but perhaps not as kind of linearly as the other 2. And one of them is the Organ on a Chip project, and particularly in this project, it's led by Dr. Linjing Zhu and Dr. Frida Passan, and they are looking at vascular devices and thrombotic thrombosis in their project. And there's also a project that's looking at nanosensing air quality to look out for airborne pathogens, which I think you can probably see what inspired that project. And that one's led by Professor Corinne Keogh and Professor Antonio Tricoli. So they're 2 new health-based nano grand challenges. There's also the nano health clusters, which are exploring lots of different areas. And I won't take too much more time to talk about that, but please go and have a look at the website and feel really welcome to engage with us or to drop myself, Zdenka or Wojciech a line if you'd like to know more. We'd love to welcome you to future events. Are there any other questions from the audience? Because I have definitely I have a few more. I've got one for Wojciech that I've been wanting to ask for a while, actually, if there's no other questions, but I don't want to take space from other people. I was wondering, Wojciech, I'm going to go for it. I love hearing about the models that you're building in your team. I think it's really exciting, and I hadn't put together the idea of linking the gut with the lung model in previous talks. So I'm excited to read a bit more about that and to learn more about that. But I was wondering, how far away do you think we are from not needing the baboon model at the end? How far away are we from having robust and reliable methods where we don't need to rely on animal models?
So this is a really good question. So I'll address maybe very quickly the lung and gut. In human environment, we actually have the lung and gut axis, and this is the major one which is impacting on the lung health. It's actually more complex because it's a brain, lung, gut axis. And now how far we are from this whole endeavor. A few years ago, DARPA put $70-odd million to develop multi-organ on a chip. They still call it that way, but actually it's exactly the same platform which we use in the lab. I can say one thing. FDA at the moment introduced precisely the same system which we have in the lab in the pipeline for testing and examination of materials. They are super interested. I was very pleased actually to see that they registered for my seminar tomorrow. AstraZeneca also registered. So how far we are, it is difficult to say. I think we are very close that the regulatory agencies will accept this, let's call it now very broadly, organ on a chip as the one of the assays for testing, optimization, and the screening of the materials. I think we are close in some of the conditions that the FDA, TGA will be accepting those models. The reason is very simple. We cannot create this disease in any of these animals. And whether we will have to have a baboons, I would still at this stage say for the next 5-10 years, yes, but we will definitely reduce the number which is necessary to be used, because we'll optimize our formulations treatments in a high throughput or semi-high throughput modes and get with a very high confidence to the baboons for testing. There are actually some treatments which people go directly to humans after convincing FDA that their formulation cannot be tested on any of the animals. So maybe exploiting this path, we can say, look, we actually have a very robust human physiology mimicking model. Why do we have to do the animal, which represents nothing that we are, as the humans?
Yeah, I mean, it'd be fantastic to move beyond the requirement for animal testing, and it's lovely to see work progressing in that direction, Wojciech.
Alice, last year I was attending a conference. This is a world largest conference in this, let's call it human replacement models, animal replacement models. There were over, I think, 9,000 people attending. Every single Johnson & Johnson, AstraZeneca, blah, 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 all the pharmaceutical companies which are doing cosmetics and all these things, every company was there and presenting. Because for them, testing cosmeceuticals or cosmetics, they don't want to do it on animals. They want to have a robust model which they can test it quickly and effectively. And for them, those kind of models are must go. I personally think it will become a gold standard at a certain point, and I hope it will happen soon.
Thank you so much. So there's time for one more question if somebody wanted to ask one. It's been lovely to join you today. So please, if there's somebody who has something they'd like to ask, please go ahead. Otherwise, what I may do is just give Zdenka and Wojciech time to make a brief closing remark to say if there's anything that they might be looking for collaborators on, or there's anything that they would like to say as a final note following their great talks today.
Oh,
Go to
sure.
Zdenka first.
Yeah, sure. Thanks, Alice. I think maybe I would like to close off by saying, well, if anything that either I or Wojciech have said today has sparked something, even if you can't quite put your finger on it, just reach out. Don't be shy. And research is all about discovering something that you know nothing about at the moment. You don't know what's going to come out of it, and usually you have a plan. You think, I'm going to go and do this, but all these new discoveries come along the way that kind of make you pivot as well, and that's actually really exciting. But if you may already have your own research project or projects that you're looking for some additional contributions to. So it might be that might be the motivation as well to reach out. Yeah.
Thanks, Zdenka. Wojciech?
Look, I won't repeat anything what Zdenka said, and I just want to say thank you for inviting us here. It was absolute privilege to be here and present to all of you. And I hope that next time when you see Tesla or any other electrical vehicle on the street, you will remember my talk about extracellular vesicles and how they can help and make us more sustainable and maybe increase even longevity of our life.
Thanks so much, Wojciech. That's a great place to end. Thank you so much, Dale, for the kind invitation to come today. And please do check out Sydney Nano's website. I'll share that in the chat now, and feel free to be in touch. It's been lovely to join with you this lunchtime. Thank you.
Thanks, Alice, for coordinating this.
Thanks.
And
Bye.
to our speakers.
Thank you.
Thanks a lot.